Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. Our guest today is Dr. Huma Qamar, who is a physician scientist, entrepreneur, community health activist, and the chief medical officer at Ocugen, where she's been leading the charge on next-generation gene therapies for rare eye diseases. She has more than 17 years of clinical development experience across gene therapy, cell therapy, oncology, and vaccines, and she's held leadership roles at Yale, Harvard, and UPenn. I think we're going to have an amazing conversation today, so thank you very much for joining me.
Thank you so much, Patrick. It's my absolute pleasure to join your podcast today, and I'm very happy to actually converse with you and the audience today about genes, about my journey, and everything.
Amazing. You have an incredible story. My understanding is you came to the U.S. with just $100, and here you are sitting today as a leader in the space. I'd love to just hear a little bit about that story and what that was like for you. If you could take us back to that time.
Yes, if my professional and personal journey spans, I would say that it's more than 17 years. It's more than 20 plus years now altogether. And since my childhood, I just feel like I think outside the box. And not only that, I feel like there is no box to start with. So that was my thinking. And I am originally from Pakistan. I was born in Rawalpindi, that is just closer to the capital of Pakistan, Islamabad. And my upbringing has been in a very educated environment. And however, I come from the Northwest Frontier province, which is called Khyber Pakhtunkhwa, and that is the border of Afghanistan, and where the literacy rate for women was not that high. Now it's actually getting much better. So at that time, even till now, women are still striving to get into the empowerment mode. And my mother and my family, they were very supportive of that. My mother is a very educated woman, and at that time I had decided that I will do something bigger. When I was a kid, I always wanted to be a physician and always wanted to be a physician in the United States of America. I didn't know how to actually get to that. And I was a big fan of Michael Jackson. So I knew exactly Michael Jackson, and I was a diehard fan of him. And that's what I said in my visa interview to the officer, and he granted me multiple years visa. So, and also I had the first one from Pakistan to get the Yale scholarship. And I was also the pioneer in terms of introducing the clinical research in the medical field at that time in the medical school. So what I did was that during one fine day, I was with my professor of surgery. We were doing radical pancreatectomy, which is Whipple's procedure, which is the longest procedure. And I was very confused. I was stressed out. And he told me one thing: Huma, your eyes cannot see what your mind does not know. So concentrate on that. So that thing stuck with me. And I am a visionary leader with an achiever style. So what I thought was that, okay, this is the time I have to create a difference. I have to go not only for my family, for myself, but also for a lot of women who are really relying on me from Pakistan. I'm a survivor of 550 bombs during the Taliban surge, and I have always spoken my mind. I have always spoken the truth, and always for the education and innovation. So I came here, I landed here. I just had, in fact, $88 in my pocket, and I literally started from scratch there. I already knew that I'm here for good. And, I was very much motivated. I came here, I started doing my research at that time after Yale. I went to Howard, did the triple negative breast cancer research in Washington, D.C. and Georgetown. And then from there went to Harvard. And from Harvard, oncology has been the focus of my career, although my specialty was internal medicine. And what I did was that those $88, I'll tell you, I saw a movie for $10. So technically $78 were left. And that was a very motivational story, how to be in US, and that too without getting married. So I think each and every step that took me was that every morning I was going and I was talking to everybody sitting at the research facility, that this is what I bring, this is my resume. Do you want me to just help you as a volunteer? And actually one of the cardiothoracic surgeons saw me and said, do you know how to operate Macintosh? And that's how I kind of with my comments and started talking to him. And that was he realized that she's pretty great educated girl, and why not to give her a chance to be in the Yale lab. And then from Yale lab, the second lab, I got the scholarship. And then from the second lab and from the third lab, I actually moved to Harvard and moved to University of Pennsylvania. And then everything is just like history. I was also pioneer in terms of, doing pediatric neonatal research and oncology research. I was working at Children's Hospital of Philadelphia. I was also the director of the investigator-sponsored research unit at Fox Chase Cancer Center, also at University of Pennsylvania. So those are the 3 Ivy Leagues that I've done. And I would say that, what I felt is this: you have to be persistent and perseverance, and also having a positive mindset is what will take you where you are. And one other thing I would say that was the key to my success was, I think I had a lot of confidence in myself, and I said I can do it. So whenever you manifest in the morning, you wake up and say I can do it, that's how you do it.
Amazing. What were some of the hard moments along the way after you got to the US? And, were there some moments that you weren't sure if you can do it, and how did you get through those and propel you to where you are today?
Yes, that's a great question. And actually, nobody has asked that question to me. And I think that's one of phenomenal questions. When I got here, there were moments that you are in a different culture, in a different environment altogether. US is the land of opportunities. How are you going to avail the opportunity is very different. So you need to have some common sense and also know your strength and skills. So what I did was that first of all, I sat back one day and I said why things are, because that's what Albert Einstein says as well, that you cannot expect the same results, different results, if you're doing the same thing over and over again. So what I did was that I literally, like a strategist, put everything on the paper. And I still remember that I was sitting outside with my coffee, and I was thinking how the rent is going to be paid and what's going to happen, and should I go back? What should I do? And there was this beautiful lady who was sitting next to me in Washington, D.C., and she had lost her son. And her son was a veteran in war. And then, I was a soldier in war. And then she was telling me her life story and everything, and she was very confident and how she was dealing with stuff. I think that positivity that day actually changed my life. And what I knew is that you need to think 10 years ahead of you, and you need to see what resources you have. And that has exactly impacted my personal growth and also the growth of all the other individuals as well. So what I did was that at that time I really looked at the US healthcare organizational delivery, where US is going, what are the skills that I have, and how I can integrate into the system. And one thing I learned is I had one thing, a positive thing in me, was networking. I like to talk to people. I like to make friends. I have been involved in political health policy side as well in the past. So that's what I started kind of like utilizing. And I think it was word of mouth. And till from then to now, most of my jobs have been based on the word of mouth, and I did not have to apply for the job. So that tells you that those were the great skills and networking skills and, the confidence that got me to the next level. However, there were hard moments. But I think the only hard thing that helped me was my hard work, which is a check that can be cashed anywhere.
Speaking of hard work, we're in a challenging time for biotech and gene therapy in particular, but you all seem to be making it work. We're recording this on the 30th of May, and just a day or two ago, I think you got pediatric breakthrough designation for one of your gene therapies. I'd love if you could talk a little bit about what you're working on today, what's challenging, but also some of the success that you're seeing, because clearly you're finding a way to pave the way through it.
Yes, and that is a great achievement for Ocugen. Ocugen is a patient biocentric company, which we are a leader in. Our mission and vision is to cure blindness diseases such as retinitis pigmentosa, Stargardt disease, geographic atrophy secondary to age-related macular degeneration. What this RPD means is for Stargardt. Stargardt is really an unmet medical need. Starts right from childhood. There is actually no approved product for these patients across US or globally. So what does it mean is that these patients would potentially go blind earlier in their life or maybe mid in their life if there is no treatment. There are many trials that have been in the past, but somehow they have not progressed it to the next level. And it's very hard in the regulatory framework for FDA that the most important thing from the clinical side and regulatory side is that not only from the preclinical side, we have to make sure that all preclinical data is aligned. We go through phase 1, 2, and 3. Now, the most important thing is this, that this pediatric population, which is the unmet medical need, 40,000 people alone in US and almost 100,000 in Europe as well, if you combine US and Europe. So what does it mean for the patients is this, that we have recently also got alignment on phase 2/3 pivotal confirmatory trial, which means that within a year we will be for the BLA and approval. And that is a hope for these patients. And
Yeah.
I do get these requests all the time where their parents are calling and saying that, how devastating is that word blindness for their kids, for their loved ones, and for their family members. So we have done that. We have testimonials of one or two patients where their functional vision was restored. And this OCU410, I would say ST, is a RORA gene, very powerful gene that works on a unique four-way mechanism of action, which is complement system, working on the lipid metabolism, oxidative stress, and reducing inflammation. So that is where we come into play, and which is a one and done treatment for life with a single subretinal injection.
Gene therapy in the eye is in a kind of nice sweet spot for where the technology is today, right? Because you need a lot less vector than almost any other organ because it's so small. It's also pretty protected from the immune system in a way that most other organs are not, right? Are there other advantages to gene therapy in the eye that I'm not thinking of? And also, what are the disadvantages, if there are any, or the challenges of what you're doing in the eye?
Yes, so that's a good question. So in terms of depends on what kind of vector. So for example, for our product, it's AAV5, which means it's a fully capsid vector. And also when the delivery is through single subretinal injection in the center of the macula, in the temporal infratemporal region, and also we also our surgical procedures that we have standardized throughout our gene therapy programs is to not detach the fovea, which is great. And what happens with this, the major advantage is this, that within 6 months you start seeing the functional vision getting improved, and also geographic atrophy. If you look at it in terms of Stargardt, we have released the data from the baseline was 54%, which is pretty cool. It's trending in the right direction, and the safety and tolerability is incredible with no serious adverse events related to the product. So safety becomes the main issue here. And also with gene therapy, inflammation is the hallmark of all of those gene therapies. So having a regimen of prophylactic pre and post medication is important. Also, standardizing your surgical procedure is the key. So I'll tell you, I've said it in numerous forums that surgeons make or break the study here,
Mm.
vitreoretinal surgeons, because retina is very different. And we are not only targeting late stage, but we are targeting early to intermediate to advanced cases. So there is the purpose of this gene therapy is twofold: either to stabilize whatever the photoreceptors they have and the vision they have so that they don't lose more, or restore back or improvement, which we have seen both. So these are the challenges from the surgical perspective. Also, with subretinal injection, it is, any complications can happen, but that also depends that how you're training your surgeons, training your sites. That's a lot of work that we do from our end, and some of the key learnings that we have, and also adjusting the volume and the concentration based on your preclinical and the phase 1 data that comes in. How are you going to do that? And the best thing is all our programs have only one dose final for phase 3, which actually for Stargardt as well. And that is where we have optimal levels there to achieve the desired result. In terms of one last thing I would mention here is that the main benefit of this is as a one and done treatment, which means that 6 months, 12 months, and moving on, 18 months, this will be for life and it will potentially improve. So you don't need another treatment after this if it works.
Yeah, amazing. What is different about gene therapy trials compared to some of the other trials you run? You mentioned, spending a lot of time in oncology. What have you had to rethink or reimagine as you've got into this different class of modality?
Yes, so great question. So I think you're the first one who has asked it, and all great questions, by the way, Patrick. So the oncology world is a little bit forgiving in terms of we have some clear NCCN guidelines. We have some resist criteria that we go through. We also know that we can get expanded access programs easily, single patient INDs easily. We have also seen that, it's a little bit, even from the budgetary perspective, from the market perspective, cancer, like curing cancer is one of the key goals of our health budget, and we are there. However, we have not really gotten success into major therapeutic areas, but we are making strides and we have been successful. So with oncology, you have a little bit flexibility in the protocol. The only thing is this: the protocols are also very complex. You can combine the standard of care, you can adjust the doses, and also kind of I would say that from the discretion of the investigator, a little bit more discretion is available. In terms of the CTCAE criteria for adverse events, we have a very good classification that happens, which is present for the oncology. So anything that we are classifying, we already know the roadmap to that. It is very well defined in terms of metastasis, in terms of resist criteria, everything. But when it comes to gene therapy in eye and ophthalmology, I'm going to say, and overall gene therapies are hard, but in terms of ophthalmology, depending on the condition, you will be surprised that the safety reporting criteria is CTCA criteria, which is systemic criteria. So there is no ocular dictionary that actually will give you exactly how to classify that. So it's also discretionary by the investigator, but also it becomes a responsibility of the sponsor to carefully look at it with the help of their medical team, what's going on. A little bit of an example. For example, adverse events of special interest, anterior uveitis. I mean, this is, if anybody is listening to this, they can attest to that the regulatory agency still has to come up with that dictionary to actually work on that, because there's so many things that are different. And if you talk about retinitis pigmentosa due to genetic mutations, there is a lot of heterogeneity. Also, inherited retinal diseases are different to present its symptoms differently in different population, different ethnicity, and also the clinical and or genetic diagnosis, both syndromic and non-syndromic, is way different. So what we have learned is that it is based on experience, A. B, as ophthalmology seems very easy, it's not. C, the surgical techniques are very, very complex. Fourth, we do not have a robust system for safety reporting or looking at efficacy in terms of it's like a one size will not fit all. So that has to do a lot of thinking. The last but not the least, and anybody who is listening to this will attest that, is that the patient reported outcomes are very well defined in oncology. The patient reported outcomes in this field is somehow not validated. There are few common questions that we go through, and it's still subjective. So still, I think there is some need around that, and I think people will attest that it's hard to kind of even if there is a PGI score, or if it's an NEI visual function questionnaire 25, so it just cover, but somehow it's not there as oncology is great.
Right. You mentioned this challenge of heterogeneity and different genetic subtypes. I'd love to hear a little bit more about what the current state of the art is and where the challenges are in patients getting a genetic diagnosis and other ways to identify this as early as possible. Because you mentioned having not just very advanced cases, but also early and intermediate. And I would guess that's where you're going to have the most effective gene therapy is if you can catch it as early as possible. So I'd love to hear a little bit about that landscape and what challenges there are.
Yes, so that's a good question. So for example, let's talk about RP first. I'll talk in very simple terms. Luxturna is the only approved product for retinitis pigmentosa gene therapy only for RPE65 mutation. So in the RP landscape, RPE65 genetic mutation only contributes to 1% or less landscape.
Wow.
So the rest of the 98 to 99% are without the active treatment. And if you look at it with the recent XLRP GNJ trial that has failed, was a potential hope for the patients for 6 to 8,000 patients, but still that's not there. So what is there is that Ocugen has a novel modifier gene therapy platform, and I can explain that in a little bit later. What does it mean is that it's a total gene-agnostic platform, a total gene-agnostic limelight trial that we are conducting, which means we're going for BLA next year, in mid next year, 2026. Now, what does it mean for the patients? Because there is a lot of heterogeneity. If RPE65 for Luxturna took years, and 5 to 7 years back, there were almost 10 to 12 companies which were in the business of these gene therapies, and they could not make it to the phase 3, which means it's a lot of scrutinization. It's a lot of, like, I would say, validation required from the agency to get to that level. And the data has to be that effective in terms of not only safety tolerability, but also durability and efficacy is the thing. So we have recently released our durability 2-year data on our 400 101 study, and the same product is now in phase 3. But let me tell you, heterogeneity is, you cannot have, there are more than 150 genes associated with RP. There are more than 10% of the population for RP that have clinical diagnosis, but no genetic mutation associated with it. What happens to those patients? I have talked to those patients personally myself. They feel helpless. They are losing their vision every day. And that's where the hope is for the 400, which also is under the expanded access program approved by FDA, which is the first time for a company like this to get approval. So what happens is this, all the genetic mutations that we have, it's very difficult to have one product for each genetic mutation. You need to have some sort of thing. It's not your regular gene editing, gene replacement. It's novel gene modifier approach, which means it resets the homeostasis and improves the outer nuclear layer, and also improves the phototransduction, circadian rhythm, metabolism, all of the functions that are associated with that. And as the inventor of the program states, and I always like to quote in my, in my interviews, that one bad apple spoils the bunch. It's like a circuit. So these master gene regulators are part of our novel modified gene therapy platform, which means NR2E3 delivery or Aurora. These are master gene regulators, or like a master key to the system to fix that. Like, how you have a laptop, you shut it down, and then, you restart it. And then just that's the whole thing. That's a whole, I would say in layman terms, how it works.
Yeah, I like that analogy. So if I play that back to make sure I'm getting it, you said there's more than 150 genes that can cause RP, and clearly there's some that are maybe not discovered yet because they're even rarer, and we have 10% or more of people that have RP but don't have a genetic diagnosis. Your therapy, rather than having a separate gene therapy to go after all 150 one by one, you have, I really like this analogy of you turn the laptop off, or you push that hard reset button. What is that hard reset button? Is there, what is the kind of mechanism behind that, if you're able to?
Master gene regulator. It's actually inserting, which is the nuclear hormone receptors. Now, nuclear hormone receptors are the master regulators.
Yeah.
They actually control multiple functions like reproduction, inflammation, circadian rhythm, metabolism, phototransduction, all of that. And they improve the photoreceptor once it's injected. The mechanism of OCU400 is that it improves the outer nuclear layer and the photoreceptor survival. Now, mind that the photoreceptors are not dead, they're degenerated, or they're non-functional. So as long as they have some, if they're legally blind, that's a different issue, because there's nothing to form images. They're like under 20 by 200. That's a different thing. But for this one, as soon as they have a clinical diagnosis and or genetic diagnosis, and by the way, syndromic and non-syndromic, we're the only company in the world that has this thing in place for OCU400 RP. And that tells you a lot that there is hope for these patients, and we're actively recruiting for them as well, this trial. But I think the question about, shutting down and resetting is the power plug, is that button.
Yeah.
That button is the master gene regulator. When it gets inserted with the NR2E3 delivery. So remember, NR2E3 is in 400. That is our product, which is closer to the finish line for RP. And the second one is ST, Stargardt 410. It's also a gene therapy. It's a second product in the arsenal. And that is a novel modifier gene therapy with a four-way unique mechanism of action. Same thing, it's working for geographic atrophy as well. And the 410, why it is important? We have two approved products for 410 as well. That is Syfovre from Apellis and Izervay. And the challenge is in Europe, there is no approved product. They have not approved these two products over there because there is no functional benefit. That's what they didn't see. So they did good in terms of structural somehow, but the functional benefit is like if I go to, to the optometrist or the ophthalmologist and see the vision chart, and if there is no line improvement and I cannot drive or I cannot do basic things, or if I was able to see 2 steps versus 4 steps and do that, that is where the functional benefit the FDA and the regulatory agencies are looking at. So our data has been released even for 2 years for durability for LLVA was with a p-value of 0.005. So always remember, if the data is clinically meaningful and statistically significant, only then it makes to the finish line. And that's where we have consistently, all our products have not only structurally shown for GA, geographic atrophy, and Stargardt, but also for RP, truly gene agnostic in terms of functional outcomes as well. And safety and tolerability, no serious adverse events reported to the investigational product have been seen so far directly related to the IP.
So in terms of how you structure the clinical trials, is it accurate to think of it as a basket trial, or how do you structure? Maybe you could tell people a little bit how you approach this. And where the question's coming from is there a hypothesis that it's going to work for all subtypes equally, or might there be some that it's more effective? And also you have this question of early versus late. It's an interesting challenge, right, of how you think about designing it in a way to help the maximum number of patients, but also make sure that if it is more effective in some populations versus others, you're teasing that out in the clinical trial process, right?
Yes, it's a great question. And I think there is no magic answer to that, but I'll tell you what I have learned so far based on the clinical development here and in oncology as well, and cell therapy too. First of all, the preclinical data drives a lot of, information for you regarding the product, regarding the vector, regarding the delivery. That is there. It does provide you some sort of guidance. But when you're designing the trials, particularly phase 1, 2, that is where you need to not only design the trial just because a research question popped in my mind early in the morning and I was having a cup of coffee might not work. So we have to look at it has to be a multifactorial approach. What I try to tell is not only from the clinical development perspective, but also the most important thing is regulatory perspective, which actually majority of the people just miss that. And last but not the least, I always try to touch on is the business aspect, like whether you are going to take it to the market, what are the challenges? So you design away. Now, why I say regulatory, the rare diseases that I've worked with in the past in oncology as well as here have a fair chance of skipping the line and being in the fast pass lane because there is unmet medical need and regulatory agencies are willing to learn more about it and they are ready to help those patients with unmet medical need. Now, what happens is that safety and tolerability, particularly safety, is non-negotiable. So for those, I would actually advise the people, if they're doing their phase 1 trials, they should go with the classic, simple dose escalation, dose ranging portion of the study. They should just do like we did it, cohort 1, 2, 3, low, medium, and high dose. Figure out your maximum tolerable dose there, then go for the open enrollment, and then figure out if there is a dose response or not.
Yeah, that makes sense.
So in that case, I would say that has been my mantra for the clinical trials design, and that's what the company has committed to. So
yes, as we close out here, I would love if you could paint the vision or the most optimistic future that you have for gene therapy in the eye. If we fast forward 5 or 10 years and you and others that are in the field get the kind of funding and space you need to achieve some of the things you want to achieve, what do you think this could look like?
Yeah. So in terms of, 5 to 10 years from now, I'm very hopeful that at least I can talk about the products that I've been part of. And particularly OCU400, that's a hope. And I think it's going in the right direction. and a hope for all 98 to 99% of the population across the world, US, Europe, where we are covering more than 95% of the genetic mutations. The data will drive it, but I think the most important thing is that I see 5 to 10 years from now, gene therapy is the future. And particularly for the unmet medical need inherited retinal diseases, and also for geographic atrophy secondary to age-related macular degeneration.
As we wrap up here, I want to actually just ask you about something that you spend your time on outside of work. And my understanding is that besides all the hard work that you're doing in your day job at Ocugen, you also spend quite a bit of time as a language interpreter and volunteer focused around refugee and immigrant health. I'd love if you could talk a little bit about that. And I think you speak 5 or 6 or 7 languages. I am not sure how, maybe it's even in the double digits, but I'd love to hear a little bit about that because I think it's pretty amazing and speaks to your character.
Yeah, I'm a licensed certified interpreter as well as translator in quite a few languages. I speak Hindi, Urdu, English, Punjabi, Pashto, which is very hard. It's a language of Afghanistan and the northern parts of Pakistan as well. Saraiki, Hindko. I do calligraphy in Arabic as well. So that's also a fun fact. Yeah, so I do help a lot of people in terms of their immigration services. Sometimes if there is an urgency, if they need to kind of like interpret. And, I am also listed in their hospitals across here in the state of Pennsylvania and New Jersey. If there is an emergency and they cannot find an interpreter, so because I speak some rare languages like Pashto, they do reach out to me. And I'd love to help the community and particularly immigrants/refugees and help them with their immigration process if they need some sort of translation or interpretation. I would say early childhood intervention services as well. I've helped a lot of people in that. So I'm just passionate about giving back to the community. And that is one thing that I do from health side and the other side.
It's amazing. If I found myself or a family member in that kind of situation and had someone like you on the other end of the line helping out, I think I'd feel like I had a guardian angel watching over me. So I think that's amazing. As we wrap up here, I just want to say thank you for sharing all your insights and all the work you do. Is there anything that people listening can reach out and help you with? Are you looking for anything, hiring or connections, collaborations, anything like that?
Yes, I think I would actually I just want to say a couple of things. There are 2 things. First, for all the females and all the people who are watching me, that, be positive and believe in your dreams and whatever you want to achieve, because you will one day. And as I said, confidence is the key to success. The second most important thing is in terms of, the patients or the families who are watching us, the Limelight trial that we are actually enrolling patients, they should go to the right channels, follow us on the social media as well, and seek help when it is required. And they should not be discouraged by any of the diagnoses that they are getting, because there is hope. Maybe there are potential options that people should be looking into. And and anybody who wants to pursue their career in the clinical development or in the biotech side should be passionate about, not only, it's not just a paycheck, for at least for me. I'm very passionate to the mission and vision of the company and committed to the patients. And I think if they feel the same way, they should really dedicate their career, and they will definitely get the fruits of all the hard work at the right time, but they have to be patient.
Amazing. Well, thank you. What a great note to close out on. I really appreciate you taking the time today.
Yes, thank you, Patrick. Thank you for your time today.
My pleasure. And thanks everyone as always for listening, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Sanogenetics on Twitter, LinkedIn, or visit sanogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.